My name is Steven Breazzano. I'm on the Piper Jaffray Biotech team, and I am joined here by Adrien Lemoine, VP of Business Development at Orchard Therapeutics, who'll give us a brief overview of everything that's going on, and then we'll dive into Q&A. Thanks, Adrien.
Thanks, Steven. Hi, everyone. I'm going to give you a quick overview of where we are about and where we stand currently at Orchard, and then we'll entertain a few questions with Steven and for the audience. So I don't know much you know about Orchard, but we are a late-stage biotech company, which is quite original because we're quite new as well. We've been created officially May last year. We've been in stealth mode for a few more months at F Prime before that, and our focus is really on ex vivo antiviral-based gene therapy and rare diseases, trying to bring, if we can, curative solutions to the terrible genetic conditions where ex vivo gene therapy has a good chance of making a profound effect. We're currently focusing on two main family of disease: primary immunodeficiency on the one hand, where, as you will know, there's a very strong body of evidence that this approach does work and does provide profound curative effect, and also inherited metabolic disorders on the basis of great work pioneered in Milan, at TGet, or at Bluebird and Paris on ALD, that you can actually cross the blood-brain barrier with this modality and go about disease which have limited therapeutic options to express or to have protein getting into the brain and cross-correcting neurons. We are working with a network of partners that we are very proud of. So on the science side, Orchard was actually created by the common vision of some of our scientific founders, UCLA and with Professor Don Cohn, BCH here in Boston with David Williams, Bobby Gaspar and Adrien at UCL, the team in Manchester with Brian Bigger, Rob Wynn in particular. So all of these pioneers of ex vivo gene therapy got together a couple of years ago and decided that, well, it's time now to find an outfit to help them bring what they've been working in the clinic for the past 5, 6, 7 years into patients. And that's really where Orchard is bringing an expertise. It's on the CMC side, on the one hand, how to transfer what was done on a hospital bench for many years into a process that's approvable by the FDA and the EMA, and also then obviously on the commercial and regulatory side to bring this product to file and then eventually commercialize them. We're also working with a network of technical partners, so we are very pleased having tied recently partnership with, on the one hand, Oxford Biomedica in Oxford, UK, for the making of lentiviral vector, and with Pharmacell in the Netherlands, who will look after the transduction in a commercial setting for the patient's own cells. Currently, we've raised about $30 million in our Series A, closed last year, and as we speak, we are in the middle of our Series B. Things are going well, and we hope we can announce to the field a successful completion, which will take us probably to the file of our lead program in the US at least, 3 program post-human POC, and hopefully a rich and deep pipeline in preclinical. That's the whole team saying a little hello from our London offices. A quick word about the pipeline I wanted to mention. So our lead program 101 is for ADA SCID. We're completing as we speak the clinical development or gearing up towards a BLA file in the US. We have a program in MPS IIIA, which is currently in preclinical development at Manchester, and are doing everything we can to get to patients as fast as possible. And there's a few more programs that I hope will be in a position to announce in the next few months, both in primary immunodeficiencies on the one end and in metabolic disorders on the other end. We really believe that there's a lot of synergies, obviously, in these two families of disease, and to aggregate program under this umbrella should help us accelerate development so we can get from the early stage discovery to the patients in the most effective manner as possible. Thank you.
Thank you, Adrien. Maybe first on the ADA SCID program. I think in a lot of people's minds, Strimvelis is a pretty good drug overall in terms of the safety and efficacy profile. But maybe just walk us through where you see room for differentiation and how does the profile of your program differ?
Sure. So it is a good drug. I spent 5 years at GSK and Reggie's team, and me and a few colleagues from Orchard actually did in-license that program from TJet originally. So we're quite proud of where it's been and the work that GSK has done, bringing a purely academic pivotal trial to a file and to an approved medicine. So there's no doubt it's a great program which works. I mean, the patients are technically cured. Where we think we can bring differentiation, and that's again pending our full body of data to be published, on 3 aspects. The first one is on efficacy. So we believe by using lentivirus vector as opposed to gamma retrovirus, we trigger a more profound and a faster reconstitution of all the B and T cell compartments. And that's in particular illustrated if you compare the data to usage of ERT. So there is an ERT for ADS kid available called Adrien, which work well for stabilizing the patients. But out of the 33 patients we talked about at the ESGCT last year, so there's no publication, but we communicated the result in a congress, only one of them had to go back to ERT, when for Trimvelis, it's about 73% had to go back to ERT after treatment. Same picture for the use of IVIG, which normally if you have a fully reconstituted B cell compartment, you don't need a complementation with IVIG. In our data shed so far, only one patient had to do that, and a bit more for Trimvelis. There's a very key point as well then on safety. So we obviously touching wood and haven't seen any leukemic events with Trimvelis, but as you will know, the gamma retro field is somewhat littered with cases of leukemia on Wiskott-Levy syndrome, on XKID, on CGD. And for some reason, it doesn't seem to trigger any ADS kid, but it's still the technical overhang that one day this could happen when lentivirus, over the hundreds of patients treated across the world, across indications, from all the companies in the field, no one has ever seen any of these. Then there is the few cases of autoimmunity seen in Trimvelis that we haven't. And because we cryopreserve our product, which not only enable patients to be treated wherever they are, as opposed to have to travel to Milan, which is currently the only setting in which you can get Trimvelis, you can condition the patient, give them chemotherapy only if the drug product you've created is good enough, making sure your VCN is good enough, making sure you have enough cells. Whereas you have only a few hours with Trimvelis to inject the fresh product back into the patient, and you can't do any of these controls. So that would be the main differentiation. And of course, as I say, the fact that you don't need to go to Milan. We treat patients above 10 years of age when Trimvelis has only been ever used on pediatrics. And the stay at hospital is also shorter. Our patients have stayed between 3 to 30 days, when for Trimvelis it's been from 30 to 100 and something. So it's also proof of a faster reconstitution of the immune system and the patient and the family being able to be discharged and come back home much faster.
Great, and maybe just walk us through the data that you guys have published. How many patients, how long have they been followed, and what have been some of the key infection rates?
So we had to publish a full and updated data set. So as I said, last year we presented the data set on 33 patients across 2 sites, UCLA in LA and UCL in London, with, in our eyes, very, very good data. I mean, survival 100%. So if you compare that with obviously untreated patient, but even patient treated with a non-matching or a match unrelated bone marrow donor, it's a significant improvement. As I said, use of ERT, only one patient, IVIG, only a couple. So it's really, really strong data with a follow-up ranging from, so back then it was between a few months and 4 years. As of today, we have 47 patients being treated across these 2 sites, and the follow-up is between 0 and 5 years and a half. And yeah, everything is going very well, which makes us very confident approaching the regulators in view of preparation of the filings.
And maybe switching to commercial, I think a lot in the field believe that Trimvelis was priced maybe too low, for a commercial product to be a standalone, and I think GSK at conferences has almost said as much. How do you guys think about the value proposition of your product, just given the benchmark out there?
I think like for any other competition situation, you have to rely on differentiation. And we, as I said, we're fairly confident. We have quite a few good arguments for differentiation, which translate into cost efficiency. ERT is a very expensive treatment, it's to the tune of a few €100,000 or dollar per patient per year, depending on the patient age. So if you can avoid that cost in a significant manner, we believe payers will be considering favorably the value we bring on the table. And then it will be, of course, dependent on negotiation and how we can make the case that there could be a different pricing in place for OTL-101 compared to Strimvelis. But again, we're confident there's the price set by Strimvelis, and as you say, because GSK openly makes no mystery that it's barely a break-even price, I think it doesn't necessarily put a damper on the whole field. I want to hope that there's room for more, because these developments are still pretty expensive, and the manufacturing of these drugs are very expensive. So you need to be able, obviously, to recover your cost and keep on investing back into R&D.
Shifting gears to MPS IIIA, Sanfilippo A. Historically, this hasn't been a disease that has responded well to transplant with the CNS manifestations. Maybe just walk us through why you think a lentivirus-based approach can be impactful, and how you selected this disease.
So to me, it's the same mechanism that underpins the success that we've seen so far with MLD at GSK TGet on the one hand, and ALD with Bluebird, which are two indications for which bone marrow transplant doesn't do much at all, is the fact that you can really turbocharge the expression, and you can drive supernormal expression of the deficient enzyme in the brain, when bone marrow transplant can only achieve wild type of expression. So that's the key pivotal element, and by the choice of the right promoter driving the gene expression, you can really get to a level of protein secreted in the brain that will base the brain and be taken up by the neurons and achieve the correction we've seen in MLD and ALD. So that's really the premise of our reason to believe that MPS IIIA, which shares some features with these two devastating CNS conditions, could be a success with ex vivo gene therapy. And for now, our preclinical data look pretty stellar as far as I'm concerned, both on the expression of enzyme in the brain of mice and also on impact on behaviors, which there's a good mice model in MPS IIIA, and we have pretty good data published from Brian Bigger and his team that demonstrate that. So we're trying to get now to humans as fast as we can.
Maybe any questions from the audience? Maybe another one. You guys have treated 47 patients in ADA SCID. Have you talked to regulators about how many they're looking for a total package, and where FDA and EMA might be, and where they're thinking about the total package?
Yeah, so our main body of evidence is patients treated with the so-called fresh cell protocol. So that's been historically implemented at UCL and UCLA, where the cell post-transduction are infused back in the patient within a matter of a few hours. So we currently have one open trial at UCLA. We're opening one at UCL shortly to explore and confirm that the fact that if you cryopreserve the cell post-transduction, the drug product is not affected and the efficacy is the same. So we're keen to bring human data, not only in vitro comparison, to make this point, and regulators do welcome that. So we keep on rolling on these two trials, but we're pretty confident that there's no need for more patients to make the point that the efficacy is what it is, and the safety as well. There's obviously going to be a 15 years follow-up of the patient, but that's a bit the rule of the game in ex vivo gene therapy, to make sure the expression is stable and the safety is maintained, or is as good as expected. But we don't think we need much more data at this point in time to go and engage BLA discussions in particular.
And ultimately, how many centers do you believe you'll need to have a presence at to capture all the patients in the US and Europe?
Well, the beauty is because you can cryopreserve the cells with what we offer, you don't need to have a physical presence to offer the therapy. The cells are harvested, sent fresh to a CMO like PharmaCell, who will do the transduction. You have a few days to do that, and it's using the same network as you have for bone marrow transplants, which are well in place for many years, and you can ship cells from Australia to North America. It's already happening for the past 20, 30 years now, easily. And then the cells are transduced, frozen, and then you have all the time in the world, more or less, to send them back to the patients wherever he is. So as long as the hospital has a BMT facility, a physician locally who knows about PIDs and can follow the patient can be treated anywhere they want, which is much better for the family because we still need to stay for a month, 2 months, and these patients are mostly kids, infants, so you need 1 or 2 carers to travel along. So being treated locally is a big benefit.
Great, thank you.
Thank you.